Hello to everyone watching and attendees at the Texas Tech University Undergraduate Research Conference. My name is Aldo Hernandez. I am a Cipher Scholar and undergraduate researcher at Dr. Gerardo Gamez's research laboratory at the Department of Chemistry and Biochemistry. Along with my co-authors, Layan, Shabaneh, Kevin Finch, and my mentor, Dr. Gerardo Gamez, today I will be showcasing our work on operating condition effects on the elemental mapping of silver nanoparticles using glow discharge optical emission spectrometry. Silver nanoparticles are amongst one of the most commercialized and consumed types of nanomaterials. These kinds of nanoparticles are prevalent in a large variety of fields. For example, they are often used in biomedicine as contrast agents for MRI scans, or as cancer treatments or imaging tools. Silver nanoparticles also have well-documented antimicrobial properties, which has promoted their use in water treatment, food packaging, and even textiles. Figure 1 here shows some of the broad silver nanoparticle utilities to different extents. As time progresses and we find more and more applications for these materials, the more we must produce. This is most highlightable in silver nanoparticles due to their popularity, though similar trends can be observed for nanomaterials in general, of different shapes, sizes, and compositions. These differences are a key factor in how nanomaterials react to their surroundings. There are estimates for rates of production on a time basis. However, we do not have a full understanding of the extent of the exposure and distribution of nanomaterials. Furthermore, the interactions between nanoparticles and ecosystems, or even living beings, is not fully realized. With this, the problem at hand starts to develop. To improve our understanding of the aforementioned materials and their interactions with their surroundings, the kind of questions we need to answer are quantitative, such as how many or how much, and qualitative, such as what the composition is or what kind. The ability to map elemental components on a measured spatial area is known as chemical imaging or elemental mapping. There are several techniques that are able to do elemental mapping on nanoparticles, though a common problem is either poor cost effectiveness or poor information throughput, which is the speed at which we can produce our emission information and measure it. We are developing glow discharge optical emission spectroscopy for nanoparticle characterization, or GDOAS for short. GDOAS is a plasma-based atomic emission technique in which we can measure the light released from the excited charged species, which is the emission, and this emission wavelength is unique to each element, and we are able to map that spectral information onto spatial areas. The key takeaway about GDOAS is the ultra-high throughput capability. It can be more than 1,000 times faster than alternative techniques. To put this into perspective, to do elemental mapping on an area of arbitrary size can take several minutes to hours to see the completion, which in comparison would take GDOAS just a few seconds to finalize. Arrays of microdeposits, or microarrays, is the formatting of our analytes. These figures show examples of microarrays used in other works and with other techniques for comparative examples. The left depicts gold deposits in 20 micrometer diameters, and the right depicts human epithelial cells, individually enclosed in separate deposits. In principle, a microarray deposition configuration shows promise in being able to study single-cell interactions. Such ability would be useful in characterizing the nuances of individual nanoparticle behavior. This figure depicts the imaging instrument in schematic form. Our emission light originating from the glow discharge lamp is collected, and it passes through a lens that serves to collimate the light. The moving stage and mirror create the push-room component of our system, though here the slit width has been opened to fully encompass our analyte spatially. The spectrometer is operated in monochromatic imaging mode, and the bandpass filter serves to transmit selective spectral information. Notice that it coincides with our closely monitored silver emission wavelength at 328 nanometers. Depicted in the figures is the analyte preparation process. Without confinement, the surface experiences excessive wetting. We use vinyl masks to confine the nanoparticles suspended in solution. One goal was to avoid overflowing of the solution, hence the single microliter deposition sequence and drying time. This figure showcases some of the key steps of our measurements. To recap, our deposits are created, then are analyzed with atomic emission. I will point out the copper electrode, which serves to promote a beneficial electric potential at the deposit site. Figure 9b shows an active GD measurement with the deposits highlighted, and figure 9c is a 2D elemental map at 328 nanometers. So moving on to some of the results. In this method development, the parameters under which the plasma is run under, namely the power in units of wattage or pressure, have a drastic effect on our analytical ability. In order to track the effects of operating parameters on our imaging capability, we take individual emission maps of the sample and track how the emission intensity changes over time. These iterations are processed and plotted as a function of running time. Note the normally increasing peak widths at increasing pressure. Increasing peak width is indicative of slower consumption of our analytes. The inverse is true for powers. At increasing powers, more material is being atomized and emitting light. Thus, material consumption is faster, hence the narrowing peak widths. Another figure of merit we consider is the spatial resolution. In this context, you can think of spatial resolution as the minimal physical distance needed between two objects to be able to tell them apart from each other. In this work, it translates to the ability to distinguish individual deposits apart from each other. We define our spatial resolution as the length of a rising or falling edge of the cross-section of our emission. We have 3 deposits for replicates and 2 edges per deposit, for a total of 6 edges. We only use the inner 4 edges as the outer ones fail to reach a representative baseline. Note the improving spatial resolution as a function of pressure. It is important to point out that these resolutions are only relative to each other and not absolute distance measurements. To report the figure of merit in absolute units, we require the assumption of homogeneous or an even distribution of the analyte to be representative. However, we are not making that assumption because it is likely not the case. I want to come back to this figure to reinforce one of my previous points. If you look at figure 9c, I will point out the differences in brightness of the spots, not only from spot to spot, but also within the same spot. This may be a little bit elusive to the possibility of uneven nanoparticle distribution. The final figure of merit presented here is the limit of detection, which is defined in this work as the background equivalent concentration, or BEC. Our LOD denotes what the minimum amount of mass we need to be able to distinguish it as a silver emission source. This figure showcases the LOD at comparable powers at each of the pressures. The LOD improves, i.e. we can define fewer and fewer quantities of mass as pressure increases. The plot on the right showcases the values the LOD is dependent on to better monitor the effects of the parameters on the specific criteria. So just to offer a couple of summarizing points. Emission intensity increases with power and with decreases in pressure, and vice versa. Increased emission intensity is indicative of increased sputtering rates. Our spatial resolution improves with incremental pressures, and our relative error is decreasing faster than our single to background ratio, allowing us to improve our limit of detection at incremental pressures. Another key takeaway point that I want to present is that our LOD is already within single cell nanoparticle interaction studies, and we can do this with just a few seconds needed for acquisition time. As previously mentioned, we are currently in the process of determining silver nanoparticle analyte distribution in our samples to have absolute spatial resolution values. However, we also want to study the effect of different substrates. These fall into 2 general categories of conductive and non-conductive. A slide of glass composition will act differently than a conductive slide, for example. We also want to study the effect of differing nanoparticle size on the emission. One hypothesis is that nanoparticles become more difficult to atomize as they become more massive. We would like to see if this can be reflected in our peak widths. This could open the possibility of distinguishing nanoparticles of differing size with this technique. We would also like to characterize nanoparticles of different compositions. Gold is an intuitive successor, but we could also characterize nanoparticles with other, more material-dependent properties. Please consider reviewing some of the work of my lab mates, including our work in pristine plant tissue analysis using GDOS elemental mapping presented by one of my co-authors, Layan Shabaneh. This work has foci based on nanoparticle interactions relevant to environmental studies and in agriculture. I would like to acknowledge Dr. Gerardo Gamez's research laboratory at the Department of Chemistry and Biochemistry. I would also like to acknowledge the Department of Chemistry and Biochemistry Machine Shop and the amazing support from the Undergraduate Research Program at the Center for the Integration of STEM Education and Research, or CISER, and the Center for Transformative Undergraduate Experiences at Texas Tech University, or TRU. This marks the completion of our presentation. Thank you so much for your time.